Good morning. So for those of you who either can't see me or don't know me, I'm Eric Weiner, and I'm really pleased to be here to introduce Kathy Wu. This is the inaugural lecture of what we hope will be a new series. And many of us in the Cancer Center spent a lot of time thinking about how we want to do conferences, and we looked at attendance and we looked at who goes to what, and ultimately came to the decision that Grand Rounds as it was, which is now trying to be in person as much as possible, was largely attended by clinically oriented people and population scientists and people who were otherwise looking for lunch and or breakfast. And that there was really a need for a conference that focused a bit more on translational and basic questions. And so after some thought, a small committee of people that included Katie Politi and Megan King came up with the idea of trying a conference like this on a monthly basis. And this is the first of those. So I'm really pleased to have Kathy Wu here. I've known Kathy for many years. She was a fellow at Dana-Farber and of course is still at Dana-Farber when I was a substantially younger attending. And in fact, we worked together in clinic. Yes, briefly, she dabbled a little bit in seeing a patient with breast cancer or one or two. And so I've known Kathy now for 20 plus years. And Kathy has built really a phenomenal career at Dana-Farber. Her own interests are broad. I learned last night something I didn't know before, which is that she even had an interest in sickle cell disease and therapeutic approaches to sickle cell disease way back when, but ultimately decided that some amount of focus was needed. And her interests have really focused on immunotherapy and CLL, and beyond that, the development of vaccines and tumor specific vaccines. She is presently a professor of medicine at Harvard Medical School and the chief of the division of, let me see if I get this right, stem cell transplantation and cellular therapies at the Dana-Farber Cancer Institute. So it's really a pleasure to have you here. We're all looking forward to your talk on largely on CLL. And thanks so much for coming. We had, I will just say that a small group of us had a great dinner with Kathy last night. And in addition to being a great scientist, she's also just a delightful person to have dinner with.
Well, it's really an honor to be here, and Happy New Year, happy snow day. Thank you, everyone in the room for trudging in. It's really great to see you in person, and also to all the folks out in Zoom. I hope this is a successful series because I do think that the intersection between the clinical and the basic and really kind of being able to look at the translational opportunities that are afforded by the patients that we treat in the study are immense and very rewarding. So, and as Eric said, I do have many different interests. I think that's a hallmark of a happy MD. So like we're interested in a lot of things, and thank you for giving me the opportunity to maybe share some of the work that we've been doing in CLL genomics. Okay, so we'll start. Let's see. There we go. Disclosure slide. Okay, I thought I'd start here, which is, I think just a challenge to all of us in the cancer community, whether or not we study CLL or not, is really the challenge of tumor heterogeneity and evolution. This has really been kind of understood for quite some time now. made ever more clear through all the genomic studies that have been out there. But we know for sure that cancer is a heterogeneous population. For better or for worse, unfortunately, by the time that we are diagnosing patients with cancer, we're really here at the time of escape, where there's already so many different resistance mechanisms that have really come into play that make the tumor fit to expand and grow in the patient host. We also increasingly know that this is not happening in a vacuum, that there's an interaction with the host immune system. But again, by the time that we're seeing patients, there's so many different immune-based escape mechanisms that are at play as well. And so a lot of the questions that I think as a field that we're really interested in asking is not only this question of tumor heterogeneity and evolution, but also how do we understand these heterogeneous tumor microenvironments? Are our T cells there at the right place, at the right time? How are we responding to diverse immunotherapies? And then what is the role of tumor antigen in shaping the tumor response? I'm not going to talk today so much until the very, very end, but this is a very large area of interest in my group. And as I said, I'm going to focus on chronic lymphocytic leukemia, which honestly, the questions that I'm asking could be in any sort of tumor system, but CLL really has a lot of very unique features about the disease that have made it exceptional for the study of genomics. First, in a small tube of blood, you have very pure tumor that is readily accessible directly from the patients. The other thing is, for a cancer, it's quite indolent. And so what that means is that we really have really long disease histories of patients. We can really take snapshots in time and study evolution in real time along with the patient. And so for some time now, our group, together with colleagues in the Boston area, we've actually had this program where we've been trying to study the link from genome to phenome. How can we genomically characterize CLL? How can we understand the clinical course and response to therapy? And then how can we also functionally characterize the pathway dependencies and really thinking about how we can do better? So what I'm going to talk about today is update the group on recent genomic studies and CLL driver discovery. A bit on our efforts in looking at tumor heterogeneity in our CLL GEM models, and then just a few perspectives of where we're going next. In terms of the genomics, again, as I said, this is a very, in general, for cancer, an indolent disease. It's typically marked initially by what we call watch and wait. So there can be a long lead time, but ultimately with treatment, there can be cycles of recurrence that happen with shorter and shorter intervals, much like what we see in other tumors. I think a question that has always fascinated people in this field is, how do we understand who progresses faster or slower? And what I mean by that is that there are some patients who succumb to their disease within 2 years. There are others that can have a little bit of therapy here and there, go on for more than 10, 15 years. So why is that? What are the differences between the patients, despite all their cells looking relatively similar under the microscope? And so since forever, there has been a long effort to try to understand those markers that we could use to distinguish amongst patients, initially looking at clinical features, protein markers, but I would say over the past 10, 15 years, since there's been the next generation sequencing that's been available to us, there's really been an explosion of knowledge in terms of the genetic alterations, later on top of that, the transcriptional alterations, and even the epigenetic alterations, so that we can understand what's going on. This slide really summarizes a lot of work that has been done since next generation sequencing has come upon us. I would say that the first studies in genomics arrived around 2010, 2011. We were among the first to describe mutated SF3B1, so a splicing factor that kind of came out of the sequencing. No one had until then kind of put altered splicing and lymphoid malignancies together. There's been large-scale studies in looking at clonal evolution. So again, CLL was one of the first places that studied really this kind of concept of clonally evolving subpopulations. And then, and you can see initially our studies were about 100 patients, and then around 2015, about 500 patients per cohort. What I'm going to describe for you now is our recent work trying to put together all of these different studies together so that we could get a cohort of more than 1,000. I want to say that during this time that we've kind of performed these sort of genomic studies, there has been vast changes in the therapeutic landscape of CLL therapy. So whereas previously it was very standard to get chemoimmunotherapy, I would say that in the same time frame that I'm speaking, there has been the introduction of targeted inhibitors of BCL2, of the B cell receptor signaling, and also introduction of immunotherapy. So the really big changes, as we start to think about the genomic lesions. So how do we build an integrative CLL map? Well, we joined forces between our colleagues in North America, but also with our colleagues in Spain and Germany, and together collected cases for which there was exomes, genomes, RNA sequencing, and methylation profiling. And there was a nice overlap of these different platforms in several hundreds of patients' samples that we collected. And this is a kind of a intimidating commute plot, but I think it just speaks of a number of different things. First, I want to acknowledge the young people who were the leaders of this project. It was really an international collaboration, so I had the pleasure of working with Benjamin Nisbacher and Xiao Lin and Gadi Getz's group, Computational Gurus, and then Cindy Han from Dana-Farber, awesome lymphoma-oriented fellow, and then Ferran Nadeau and Martí from the group in Spain, the Spanish CLL group in Barcelona. And then when we looked at these more than 1,000 patient samples, in fact, we were able to have greater sensitivity. In the magenta are all the new drivers that we identified. So each row is a driver alteration. Each column is a different case. And what you can see is, in fact, there is a list of recurrent alterations, but a long tail. You can see that a lot of our discovery is down here at the 1% or less level. So many different sort of driver alterations that we had greater sensitivity to identify because of the increased power of our cohort. Just to make a really beautiful long story short, we were able to double the number of CLL drivers that we were able to identify. Previously, there were about 10% of patients that we couldn't account for. There wasn't any sort of driver alteration that we could point to that was, this is the reason that they have CLL. And we've been able to close that gap so that there's only by now 3.8% that we can't account for. The two large categories of CLL that are well known in the clinical arena on the basis of their immunoglobulin locus, the mutated and unmutated IgHV, we finally had enough power to actually break those two groups apart and look at them separately. And they really look like very different diseases. They each have distinct molecular landscapes. It highlights the diverse trajectories of clonal evolution. So maybe by virtue of where you start as a B cell, maybe there's a path of different paths of least resistance that get you to where you're going to be. And what was super interesting is that, at least for the unmutated CLLs, their source of heterogeneity was genetic. There was a lot more putative drivers in this unmutated group, but in the mutated group, relatively few drivers, but a lot of transcriptional diversity. So really a different path to achieving that type of heterogeneity. And then what I want to show you is that when we looked at the expression, Benjamin was able to identify what he called EC's expression clusters. And then the nomenclature here is some of them were enriched for M for mutated, some for unmutated. And what you can see is that it actually breaks down the groups more or less based on mutated, unmutated, or by their epigenetics. But you can also see by the fact that there's two colors within each column that there was contribution from both mutated and unmutated to these different expression clusters. And one example, one thing that was really interesting is that by the yellow asterisk, we could see that certain genetic alterations actually also segregated together with these expression clusters, suggesting that they were a cohesive entity, each of these different expression cluster groups. So for example, trisomy 12, which is a very well-known cytogenetic abnormality associated with CLL, but for which there's a great heterogeneity in kind of the behavior of those trisomy 12s. They actually split out into two groups, one that's in a more predominantly unmutated group, another in a predominantly mutated group. And this maybe provides us with some understanding for why some samples with the same sort of cytogenetics might behave differently. And what was super interesting is when Benjamin started to look at these different expression cluster groups, they actually did display different clinical outcome because we had very long clinical, these were also clinically annotated samples as well. And this is just kind of the final data slide related to this, which is indeed when we kind of break down the samples based on their classical clinical group, based on the expression clusters, whether they were concordant or discordant to that classification, we could actually see differences in their clinical outcomes, suggesting that our expression cluster system was actually increasing the accuracy of what we're trying to do in terms of prognostication. So we've been really excited to, I mean, this was really a tour de force effort to bring together not only all these different groups together and their expertise, but also to layer on all of these different genomic layers to kind of identify unique molecular subtypes. And I do want to say that these studies were samples that were collected in the era of chemoimmunotherapy. We are actively trying to look now how these relate to the modern era of targeted inhibition. And we also are interested in trying to look at whether or not the different molecular subtypes have differences in therapeutic vulnerabilities. Now, I think, as we've gotten better with our therapies, we always have to kind of reckon what is the area of most unmet need. And I think right now, clinically for CLL, there are so many different therapies available, but we are still really faced with the conundrum of Richter syndrome. This is really, it's a rare, it occurs in 5 to 10% of patients with CLL, but it is a transformation of a small indolent histological type into a high-grade lymphoid malignancy. 90% have histology similar to diffuse large B-cell lymphoma. The majority are clonally unrelated. We know that because if we follow their immunoglobulin, the clonal immunoglobulin, we could see the same in the patient. Shown here is a micrograph that shows a sample where you can see the coexistence of these two entities within the same sample, and you can see the big kind of histological differences. These are the patients that we typically say, I'm so sorry, please get your affairs in order. There's really not much more that we can do for you. And it's been very difficult to understand molecularly much about this entity because there's been limitations of tissue sampling and it's really based on morphologic diagnosis. There's been a lack of markers and understanding of genetics. And for a blood-based malignancy like CLL, Richter's is really like a solid tumor. I mean, this is really, unlike what I said before, where there's ease in kind of having blood draws, here we have to get biopsies, often FFP specimens in order to study, and this has not been easy. But I would say that over the past couple years, because of the availability of all these nice genomic platforms, there's been really an explosion of new studies that have come out in the past year and a half. And at the same time, there's been modeling that's been done trying to really put our attention to how we can generate mouse models, whether they're PDXs or GEM models, to try to understand this. And there's been actually a lot of progress in understanding the genome, the genetics, looking at the epigenetics and the transcriptomics. And so what I'm going to demonstrate for you in the next couple slides is some of our efforts in this area. This is really work that was led by Erin Perry, who is now a new junior faculty member at the Dana-Farber in the lymphoma group, Roman Guiez, who's part of Philo back in France, and Ignat Leshner, who is now a junior faculty member at BU. And what we tried to do was assemble a nice paired matched cohort. So in other words, not just Richter samples in isolation, but antecedent CLL matched together with the Richters, where we could track evolution in time across these patients. This was about 50 patients that we collected samples on. I think the point of emphasis that I want to show you on the left side here is the CLL course. The green is the different lines of therapy. On the right side is the Richter's. And I want to show you that on the left side, it's years, whereas on the right side, it's months. So this kind of gives you a sense of kind of the time course of these patients. The black dots are the different samples that we collected on the CLL course. The yellow here is the Richter diagnostic sample. Unfortunately, there's a lot of red here, which is that the patients did succumb to their disease. There's a number here with black arrows that are living. For the most part, these are patients who received therapy and then went on to stem cell transplant and really did a complete overhaul. So we obtained exomes on most of these patients, also had some genomes, RNA sequencing and single cell sequencing data. But I want to point out to you that, a lot of these studies are really quite different. I think that the conundrum that we've met with Richter's is that it's really two malignancies in the same sample. So how do you pull apart the genomic contributions of one versus the other? And for that, we had to come up with a computational approach that was quite challenging, but we were able to succeed, where we really optimized the copy number analyses to deal with FFPE artifact. We had a number of different filters that allowed us to kind of increase the sensitivity of our analysis and deal with contamination, whether tumor in the normal or the reverse, as I said, the artifact from FFPE. And then we were able to put in our algorithms that allow us to identify clones and then also establish phylogeny. So at the end of the day, we were able to separate out the contributions of the CLL clones compared to the Richter's clones. And in doing so, then we could start to look at phylogeny and understand which branches were CLL versus Richter's and look across time. So again, long story short, I think one of the questions that has been asked in the field is Richter's, is it a distinct entity? Is it similar or is it different from de novo DLBCL? And here we had the advantage of being able to access older data of more than 300 samples of lymphoma that our colleague Margaret Shipp had collected. And then using those data, we performed unbiased NMF clustering. And you can see across the purple on the top that the Richter's really stand different. They're, separately from DLBCL. And so so this is clonally unrelated Richter's. So these are the few samples here do appear to be like de novo DLBCL, but the vast majority, the clonally related, stand separately. Among the Richter's itself, we were also, because of all the genomic alterations that we found, we were able to also perform unbiased clustering and discern that there's actually, it appears to be molecular subtypes within Richter's itself. And these TP53 has long been associated with Richter's, but we can see that there's different flavors. So this one here has enrichment in whole genome doubling. This group here, RS3, has co-occurrence with Notch1, also deletion 15q, which covers MGA, which affects MYC. And then RS5 also has Notch1 as well, wild type Notch1, and a lot of copy number alterations. There were also two other subtypes that did not have TP53, KRAS, SPEN, Notch1 together with trisomy 12, and also SF3B1 with EGR2. And again, these different subgroups appear to have different clinical behavior, where the ones that have TP53 seem to have worse prognosis. Now, what is the meaning of kind of trying to look at all these different genomic alterations? Well, one thing we realized is that maybe we could harness all of this and actually look to see this, whether this could help us devise a non-invasive approach to identifying Richter's and getting us to earlier detection. And it turns out that with simply ultra low pass genome sequencing, $150 a pop, you can focus on these different alterations that we identified and start to look. And in fact, we were able to see in this example, this is a patient where we could identify the Richter's alterations even close to 5 to 6 months before the actual diagnosis. So if you follow this in the blood cells have CLL, at this time point early on, and it's a very, very quiet genomic profile, whereas the plasma shows all of these different alterations that match very similarly to what was detected much later when the actual the tissue diagnosis was made. We've been able to see that in a number of different other cases. This is another, oopsie, this is another case. Well, anyway, let's see. Where the in the plasma, we were able to again find those kind of Richter's genomic alterations that was not evident in the blood cells. And finally, this is a case of a patient who went through transplant, and we were able to identify post-transplant relapse months before the actual diagnosis, and then institute therapy, and you see those alterations go away again. So I think, just to summarize this part of the talk, I would say that we've been able to actually find that the majority of Richter's does evolve from CLL subclones through acquisition of additional drivers. Clonally related Richter's is distinct from de novo DLBCL. There are molecular subtypes of Richter's that have, and these different subcategories do have prognostic significance. And then we're very excited about the cell-free DNA as a way to get us to non-invasive earlier diagnosis, because I think this could be really quite impactful for our patients. I think we're always trying to, I'm going to transition now in terms of talking about the immune microenvironment for Richter's. I think we're always trying to gain a bird's eye view of the landscape, and really the advent of single cell analyses has really been so impactful all around. This is something I put together with one of my postdoctoral fellows, where we tried to look at across the field. Single cell sequencing was named the method of the year in 2013, and then subsequently 2019, multi-omic analysis was the method of the year. CLL has had a bit of a lag time in terms of the rest of the field, but again, the easy access to material has really kind of stimulated us to start to look a little bit more closely. We've been able to apply this approach. Again, I mentioned that Richter's is an area where the therapeutic opportunities are not great, but what has caught the attention of many is that it turns out that there is a response to immune checkpoint blockade. So 42 to 65% responses to PD-1 blockade in Richter's. This is really quite remarkable, because a lot of blood B-cell malignancies do not have a great response to anti-PD-1. And so this sort of, across these many studies, this raises the question, are there determinants of response and resistance to PD-1 blockade? We were able to partner together with our colleagues at MD Anderson. Again, this is the work of Aaron Perry, where they had already started a trial where they had patients initially on nivolumab, and then after the first cycle, then ibrutinib was started, and then response assessment happened at 3 months. And so we were able to collect bone marrow samples from these patients, a number in the green that had either a partial or complete response, to patients that had progression even at the 3-month time point, and then, just for comparison, 2 CLL patients who were treated on the same trial. And what Erin did was she was able to take marrow samples from these patients, and through flow cytometry, you can see that the small cells were the CLL cells, the large cells were the Richter's, and then there was another population here which was neither, and this was the immune cells that were in the bone marrow, and then she was able to perform single cell characterization. And again, to summarize a large body of work, what was really clear is that the responders compared to the non-responders, when you started to look at all of those single cell transcriptomes, there was a kind of a cluster of cells that kind of segregated with a unique phenotype. And we called this cluster 1. It turns out it was high expression for a transcriptional factor called Hobbit, ZNF683. And as she started to look at this population, she was able to perform some functional studies and demonstrate through cut and run and various different sort of overexpression and knockout kind of analyses that ZNF683 does appear to regulate T cell pathways with activation cytotoxicity. When we started to look at the trajectories, the ZNF 683 high seemed to be a divergent pathway from terminal exhaustion. We also looked across other different solid tumor TIL settings, and it turns out that the ZNF 683 high does mark a population that's of patients that have better response to PD-1 therapy. Notably, we looked in melanoma across and other settings, and also she was also able to see that, you know, we did our analyses in the marrow, but to make it more clinically facile, could this be actually detected in the blood? And so she was able to look at independent patients who are responders or non-responders on the MD Anderson trial. And in fact, the responders have a very distinct profile in the blood T cells compared to the non-responders, where there is high expression of ZNF 683 and other cluster 1 genes as well. And so we were very proud of Erin and Camila to get this into Cancer Cell. We actually tried to for a cover. It did not work, so you will never see this published, only here in this seminar series. But we were trying to make a play on ZNF 683 and The Hobbit, and the idea that if those of you were Middle Earth aficionados or Tolkien lovers, you know, the idea that you can either take a path and get to the Valley of Death with exhaustion, or you can take a divergent pathway and end up back in the Shire happy. So that was our idea. Didn't work, whatever. So that I'm going to move on to the second set of study, second chapter, shall we say, in trying to look at function. And here, you know, in the same way that in the genetic realm, we've been able to study heterogeneity in patients, well, can we not actually generate mice that are actually faithful to the disease through the by mimicking some of these genetic alterations that we've identified? And then that provides us a platform with studying mechanism of disease and testing novel therapies. And I just want to point out that there are different flavors of models. I don't need to tell this audience or folks at Yale that, but the GEM models in general, in particular, I just want to point out, have the advantage that this is kind of in a physiologic setting. It does allow us to look at tumor evolution and also immune microenvironment analyses. And so for the past period of time, my group has really been interested in this question. Well, how do you get from a B cell? What are the kind of pathway hits that happen that gets you to CLL? And can we study some of these alterations that we spent a lot of time genomically identifying, such as SF3B1 or IKZF3 or DMT3A and so on and so forth? And can we start to look at these things? So I won't go over these past studies, only to say that it has in fact been very gratifying to generate these mouse models and to demonstrate that, yes, these putative drivers that we identified through sequencing actually generate CLL in mice. Most recently, we had a very nice study. Eliza Tenhaken generated the setting where, using CRISPR, she was able to introduce combinations of different alterations and really combinatorial study the different models of CLL and Richter's that we identified. But for today, I'm going to talk about new unpublished data where we've been focused on one of the newer drivers that we identified, RPS15, and some of the insights that we've identified there. So RPS15, what is it? It is a ribosomal protein. It's identified in 5% of CLL patients. It's enriched in patients who are relapsed following therapy. It's associated with shorter progression-free survival, and it commonly expresses with TP53. One of the things that we found interesting about RPS15 is that there does seem to be a hotspot region where a lot of the alterations happen. And so this kind of piqued our interest in trying to learn more about RPS15. I do want to put this in the context that across different cancers, there's been a lot of different ribosomal mutations that have been found. For CLL, RPS15 is the only ribosomal mutation that's been identified, but certainly across other cancers, including breast cancer, melanoma, myeloma, you see that this biology seems to be there. And ribosomopathies have been associated with a variety of different altered functions, so including DNA damage, proteasomal alteration, and metabolic rewiring. So we were interested in trying to dig a little bit deeper about this in CLL. So we used this in a similar fashion to the other mice that we've generated. We introduced one of these hotspot mutations that was intercrossed with CD19 cream mice. So this alteration is only present in B cells in the context of CD19 expression. So in B cells, we were able to generate both heterozygous and homozygous mutated mice. We also intercross also with deletion 15, TP53, so that there were also mice that had double mutations as well. And so this is just a bit of the targeting strategy. This was really studies led by an MD PhD student and currently at MGH as an intern. And then Marwan Kwok is an awesome postdoc in my group right now who's leading up on some of the functional studies. Neil Ruthin is in graduate school in the New York area for computational biology. So RPS15 mutations, we were very able through our mouse models to confirm that it does have oncogenic potential, because certainly over time we're able to identify that there is a population of RPS15 mice that do have expanded B cells. You can see this also in spleen sizes. Over time, it does take quite a bit of time, consistent with the human disease. It does take about 18 months to 2 years in order to see disease. So this is really a labor of love. But I would say that for sure with the RPS15 mutations, mutant mice, we do see onset of disease, less so with just the single mutant TP53, but with the double mutant, we also see not only CLL, but evidence of Richter's. But what was interesting is in the setting of hyperproliferation, when we look early on, there seems to be hypoproliferation. So if we measure the B cell percentages in the homozygous mice in the setting of pre-leukemia, it's actually depressed compared to wild type. So what is going on? How is this kind of hypoproliferation turning into hyper? And so to kind of gain some clues, we really focused on these pre-leukemic mice for which we collected B cells, and started off by just looking at gene expression profiling. And it became quite evident that there was quite a few different altered pathways, including cell cycle checkpoint, MYC targets, DNA repair. And looking more closely, we could see that this was related to either reduction in proliferative capacity, as well as there was increased G1 checkpoint activity after mitogenic stimulation and increased apoptosis. Now, these alterations in cell cycle could be due to cell stress. So we started to look at the question of whether or not there was changes in oxidative stress. And in fact, using a mitosox assay in our homozygous mice, we do see evidence both at baseline and with stimulation that there is increased enhanced oxidative stress, which is supported by the fact that when we used an inhibitor, so a pro-oxidant, we actually see that the RPS15 mice are more sensitive to this inhibitor than the wild type. Now, because of the cellular stress, does this actually, can this actually support acquisition of genotoxic injury? And in this case, we were able to use gamma H2AX, and we see in the homozygous mice that there is increase in gamma H2AX. And as we started to, there's a lot of Westerns that I could have shown you, but suffice it to say that through examination of the mutant mice, we do see impaired cell cycle checkpoint response to DNA damage, impaired response signaling, abrogation of ATM and CHEK2 signaling, and heightened intrinsic aberrant DNA damage response. And despite that, there's also increased proliferation signaling. So one of our highest hits in our gene expression was ZAP70, which has relevance to CLL. So we see that here. And there's also enhanced BCR signaling. So definitely a balance between different forces at play going on. Our next question was that is seeing these different sort of phenotypes, since this is a ribosomal protein, is there actually alteration, is there effects of mutant RPS15 on translation? And so we asked, could RPS15 mutation cause ribosomes to preferentially translate certain genes? Could the mutation cause the ribosomes, for example, to stall at specific protein coding sequence motifs, interrupting translation of certain genes? Or could it read through stop codons and then result in misfolded and degraded proteins? And so for this, we performed a ribosomal profiling. And when we started to look at whether or not there was evidence of differential translation efficiency, there were certainly many genes that were appeared to be have enhanced or depressed translational efficiency. And as we started to look at the pathways that were impacted, these included many of those pathways that I already talked to you about in the pre-leukemic setting. So cell cycle, MYC targets, cell cycle checkpoints, and DNA replication. And specific examples that we could see were genes that have very well known roles in these different pathways. We were able to support this kind of riboseq analysis by looking at protein expression, and we can confirm that what we saw as having depressed translation, so the GPX1, we could actually confirm at the protein level for GPX1 and OTUD4, and increased expression in PTP4A2. So that was actually very nice to see that linkages between translation and the pathways that we were impacting. When we started to look at, we were also able to see evidence not only in our murine cells, but also in a human cell line. We saw evidence of stop codon stalling. So you can see kind of a pileup here in terms of the relative position to the stop codon. But we also saw evidence of stop codon read-through. And so we do see that there's enrichment of certain codons in that kind of stop site, suggesting that this is not a random process, but there's actually motifs that are kind of guiding this process. And finally, as we started to look at the leukemic B cells, we could see upregulation of MYC targets. And I'm going to just skip over this, but only to say that as we start to go through our model of what we think is going on, we do see that in this mutated ribosomal protein, that there is evidence of altered translation through a couple of different mechanisms, that these do initially lead to hypoproliferation. There is elevated ZAP70 and BCR signaling, as well as MYC activation, but initially there's P53 mediated apoptosis and cell cycle checkpoint changes that are leading to that hyperproliferation, but that over time there's acquisition of DNA damage and genomic instability that tip the balance and get us to this state of hyperproliferation. So just to conclude this part of the talk, I'll just say that again, our new work suggests that RPS15 mutation is a CLL driver and reinforces the notion that CLL has these core pathways that are affected. So I didn't go into this, but across our different mouse models, we are seeing common pathways through different mechanisms that appear to be involved. And current ongoing work is starting to look at the immune microenvironment so that we can start to link the genotype with whether or not they're related to distinct changes in the immune microenvironment. In the final slides, I just want to say that I think that where we're going next in sort of our studies, a lot of the CLL work until now, I think across the field, has been really focused on the blood, easy access, lots of tumor there. But I think increasingly we do need to look at these specialized hematolymphoid organs where there is a specialized immune microenvironment that we can understand better. I think that there is a priority and interest in trying to go earlier in disease. So how can we understand those early events? How can we intervene early? How can we change natural history? We're only going to get there by understanding a little bit more about this earlier time. Multi-omic profiling, for sure. There's so much data out there, and how can we link them all together and kind of not have them as separate entities, but really trying to coalesce into kind of archetypes that we can understand. Spatial analysis. So our group is actively working on efforts to try to look at the architecture of lymph nodes and bone marrow to see how malignant cells are organized and also in relationship to their genotype, so their mutations, and do specific clones segregate with specific types of niches, and are they organized in certain type of neighborhoods? And finally, I touched upon with our cell-free DNA work some of the early detection. I'm going to end with the last couple slides speaking about early intervention, we hope in the future, but another big part of the work that my group does is think about cancer neoantigens. And from all the genomic studies that we've been doing, we've realized that there is the opportunity for these mutations to generate epitopes that can be recognized by T cells. I'm not going to go into this in great length, only to say that there's straightforward algorithms by now that allow us to start with the sequencing data and identify for us what those neoantigens might be. I want to say that some of our earliest work in the neoantigen field and kind of setting up these pipelines were in CLL, because that is where we had the data and all the tools to help us construct some of these first pipelines that were available. And certainly our vaccine neoantigen work that Dr. Weiner alluded to has taken our group very far afield from CLL. We've gone into the solid tumors, and we've been able to conduct some early proof of concept studies that such an approach of starting with tumor, looking for genomic alterations, and generating a personal vaccine is feasible. But I've always been super interested in trying to bring it back to CLL. And so I'm happy to say that right now we have a phase 1 study for patients with unmutated IgHV, led by Inye An and supported by Matt Davids and Jennifer Brown, to study and look at the impact of this vaccine alone, vaccine together with low-dose cyclophosphamide as a way to kind of alter the immune microenvironment and maybe address Tregs, and then also a third cohort to actually add immune checkpoint blockade together. And we already have enrolled a number of the first 3 patients. We're already seeing interesting immune responses compared to our solid tumor settings. These are patients who actively have circulating disease. So is it possible to even vaccinate and generate meaningful responses when there's leukemia that's in circulation? And the short answer, it seems like yes. So we are actually seeing very nice brisk immune responses to actually some of our patients. So I hope you stay tuned, and hopefully we'll have more to say about that in the time to come. I've tried to acknowledge folks along the way, but here's a more extensive list, and I really appreciate your attention, and thank you. Yes.
So how do you think about driver mutations, specific driver mutations related to transformation, related to potential for dedifferentiation of B cells leading to the clinical outcome? You listed a whole list of potential driver mutations. It's not clear to me what the individual driver mutations are doing and how you think about getting the answer to that question, if it is an important question.
Yeah, no, I think I skipped over a lot of stuff. And so I think one of the things that we can do when we have these driver lists is we can see whether they segregate into particular pathways. By virtue of kind of separating out those CLL versus Richter clones, we were able to kind of identify which of those drivers seemed to be CLL, which were Richters, and which were in a path on the way to transformation. And so some of those pathways that we see affected are related to MYC, for example. They're related to cell cycles, so it's not a surprise, but and metabolic rewiring as well. So there's many. So I think the drivers do help us think about the biology of what is going on, but I hope that we can also use them as ways for early detection. I don't know if this answers your question, but yeah.
I don't know if the question's online, but what do you think about the role of RGS-15 in normal CD5 B cells? So it's there.
Yes.
So the question is, what is its function in thinking about what CD5 B cells are doing?
Yeah.
In terms of maintenance of tolerance, for example, and their potential tolerant activity and where they are.
Right. So we haven't looked into that. I mean, I think we have the tools, and so we've really been focused on the mutant setting. Yeah. But I think it's a really interesting question. And I think that maybe a separate question could be like all of these different mutations that we're finding. Yes.
Same question.
Yeah.
It's very
The genes and what are their roles in normal B cells. I think you are absolutely correct. Yeah. Yes.
Yeah, I'm getting this correct. I think you said that the unmutated CLLs had a greater genetic diversity.
So yeah, so the unmutated there's a far longer list of mutated drivers in unmutated CLL.
I see. So I guess the question then is, do you think that the mechanism that's leading to the mutations of the IGH locus is unrelated to the genetic diversity that we're getting, or is there a relation to them, and how does that?
I think that's a great question. So the question is whether or not how the immunoglobulin mutational status relates to kind of the genetic diversity. So yeah, it's been understood that whether or not the CLLs have a mutated or unmutated immunoglobulin relates to their cell of origin, kind of where are they in kind of B cell development, and whether or not those kind of normal physiological mutational processes are present. So I think it does speak to the underlying biology of that cell of origin, and probably it helps us understand why there could be more mutations in these different genes compared to the unmutated. So that would be a way to put it together.
I have some questions.
Okay. Yes.
So Marcus Bosenberg asks, are there any recurrent genetic or epigenetic changes in CLL arising at later time points in RPS 15 mutant ones?
Marcus, hello. Great question. We haven't actually looked at that. I think that's a great question, and probably something I should take back to the group, and we should look. But we haven't looked at that. So thank you.
Okay, one last question. There's George Miller asks, can you comment on the role of Epstein-Barr virus in conversion of CLL to DLBCL?
I really can't comment. Yes, we have not looked at that. It's a great question, and certainly EBV does play a role in immortalization of B cell lines, but I don't have much deep thoughts about that. So my regrets.
Thank you. Well, thank you very much for visiting us.
Yes, thank you.